The arrival of a new baby is a time of immense joy. For Kyle and Nicole Muldoon, the birth of their son, KJ, was a cherished moment. They eagerly anticipated the laughter and milestones that lay ahead. However, their world soon took an unexpected and frightening turn. Shortly after his birth, KJ began to show signs that something was gravely wrong. The typical joys of new parenthood were overshadowed by concern. Doctors quickly realized that KJ was facing a serious health challenge. This marked the beginning of an extraordinary journey for the Muldoon family, a journey into the frontiers of medical science. Their hope rested on finding answers, and quickly, KJ was diagnosed with an extremely rare genetic disorder. It is called carbamoyl phosphate synthetase 1 deficiency, or CPS1 deficiency. This condition affects only about 1 in a million newborns. The genetic code within KJ's DNA carried a tiny error. This error prevented his body from producing a crucial enzyme. This enzyme is essential for processing protein. Without it, the body cannot break down protein properly. This leads to a dangerous buildup of waste products. The diagnosis was devastating. It presented an immediate and life-threatening situation for baby KJ. His parents were faced with a terrifying reality. The primary danger of CPS1 deficiency is the accumulation of ammonia in the bloodstream. Normally, the body converts ammonia, a toxic byproduct of protein metabolism, into urea. Urea is then safely excreted. In KJ's case, this vital process was broken. Toxic ammonia levels would rise rapidly. This could cause severe brain damage. It could also lead to coma or even death. Every protein-containing meal became a potential threat. Managing his condition required constant medical intervention. The future looked incredibly uncertain for this little boy. His life depended on finding a way to control the relentless rise of ammonia. Faced with this dire prognosis, KJ's parents, Kyle and Nicole, had an agonizing decision to make. The conventional treatment for severe CPS1 deficiency often involves a liver transplant. However, finding a suitable donor organ takes time. For a critically ill infant like KJ, waiting might not be an option. He might not survive long enough to receive a transplant. The alternative was a highly experimental approach. It was a gene therapy specifically designed for him. This therapy had never been tried in humans before for this condition. It was a leap into the unknown, yet it offered a glimmer of hope. Where options were scarce, they chose to pursue this pioneering path. The concept of creating a medicine for a single individual is truly revolutionary. For baby KJ, this was not just a concept, but a lifeline. Scientists embarked on an unprecedented mission. They aimed to design a treatment that targeted the unique genetic makeup of KJ's cells. This approach, known as personalized gene therapy, represents a paradigm shift in medicine. Instead of a one-size-fits-all drug, the treatment is bespoke. It is crafted with meticulous precision. The goal was to correct the specific genetic error causing his CPS1 deficiency. This was a journey into uncharted territory. It required immense skill, dedication, and a willingness to innovate rapidly. The technology at the heart of KJ's treatment was a refined CRISPR method. This method is called base editing. Unlike earlier CRISPR techniques that cut the DNA double helix, base editing works with more subtlety. It acts like a molecular pencil with an eraser. It can rewrite a single letter of the DNA code. In KJ's liver cells, a specific DNA base was incorrect. This tiny error was the root of his illness. The base editing therapy was designed to find this specific spot. It would then chemically convert the incorrect letter into the correct one. This was all done without making a disruptive cut in the DNA strand, offering a potentially safer way to edit genes. Remarkably, this custom-made therapy was developed in just 6 months. This rapid timeline was a testament to incredible scientific collaboration. The Children's Hospital of Philadelphia spearheaded the effort. They received crucial support from the National Institutes of Health. The Food and Drug Administration provided essential guidance. This allowed the experimental treatment to move forward responsibly. Several companies also played vital roles. Aquitas Therapeutics and Integrated DNA Technologies generously donated key components. This collective effort demonstrated what is possible when science, medicine, and regulatory bodies unite for a common cause. It was a race against time, and every moment counted. Many brilliant minds contributed to this groundbreaking achievement. Dr. Rebecca Ahrens-Nicklas, KJ's physician at CHOP, was central to his care. She championed the quest for a novel solution. Dr. Kiran Musunuru, a gene editing researcher at the University of Pennsylvania, brought his expertise to the table. His work was pivotal in harnessing the power of base editing for this specific case. The therapy utilized lipid nanoparticles to deliver the gene editing tools to KJ's liver cells. These components were designed to be redeliverable. This meant KJ could receive multiple infusions if needed. The success was truly a team effort, a symphony of scientific and medical dedication. The administration of KJ's personalized gene therapy was a carefully managed process. He received 3 infusions of the custom-designed treatment. Each dose delivered the base editing machinery to his liver cells. The hope was that these molecular tools would find their target. They would then correct the faulty gene responsible for CPS1 deficiency. The medical team monitored KJ closely. His parents waited with bated breath. The world of medical science watched with keen interest. This was a critical test for the potential of hyper-personalized medicine. Every small sign of progress would be a beacon of hope for KJ and for many others. After the very first dose, there were encouraging signs. KJ began to show improved tolerance to protein. This was a significant early indicator. It suggested that the therapy might be working as intended. If his liver cells could start processing protein correctly, his ammonia levels would hopefully stabilize. For his parents, this was the news they had desperately longed to hear. While it was just the beginning, it offered a profound sense of relief. The initial response provided crucial validation for the scientific approach. It fueled optimism for the subsequent stages of his treatment and recovery. The journey was far from over, but this was a vital first step. Following the subsequent doses, KJ's condition continued to improve. His doctors were able to reduce the medications he needed to control his ammonia levels. This was a major step forward. It indicated a more stable and self-sufficient metabolic function. Perhaps even more remarkably, KJ successfully weathered common childhood illnesses. He experienced a cold and a gastrointestinal infection. Crucially, he did so without the dangerous spikes in ammonia that would have previously been life-threatening. This resilience was a powerful testament to the therapy's impact. He was beginning to live a more normal infant life, less constrained by his severe condition. As of May 2025, nearly 10-month-old KJ Muldoon is developing well. He is reaching important milestones for his age. He can sit up on his own, he can roll over. He plays with the curiosity and joy of any baby his age. His physicians emphasize that it is still really early days. They caution that this is not yet considered a definitive cure. However, the progress he has made is undeniably significant. It is a source of immense hope for his family. It is also a profound inspiration for the medical and scientific community. KJ's journey illuminates the transformative potential of gene editing. KJ Muldoon's case is more than just one child's successful treatment. It is a landmark moment in the history of medicine. It signals the potential birth of what some are calling CRISPR-on-demand therapy. Geneticist Fyodor Urnov from the University of California, Berkeley stated, "This is the year when CRISPR-on-demand is truly born. This means treatments could be created for individual patients. This is especially important for those with ultra-rare diseases. These are conditions that affect so few people that traditional drug development is often not feasible." KJ's story shows a new path forward. It is a path paved with personalized genetic solutions. This groundbreaking approach highlights the power of combining several key elements. Rapid diagnostics are essential to identify these rare conditions early. Regulatory flexibility is needed to allow for innovative single-patient treatments under careful oversight. International scientific collaboration, as seen in KJ's case, pools expertise and resources. Modular CRISPR platforms, like base editing, can be adapted relatively quickly. They can target different genes for different conditions. This template, established by KJ's journey, could potentially benefit thousands of individuals. These are people battling genetic diseases that currently have no effective treatments. The hope is immense. Researchers are already working diligently to adapt base editing for other conditions. The success with CPS1 deficiency provides a powerful proof of concept. Scientists are exploring its use for genetic forms of blindness. They're investigating its potential for sickle cell anemia. Rare neurological diseases are also a focus. The list of devastating genetic disorders is long. Each one represents immense suffering for patients and their families. Dr. Joni Rutter, director of the NIH's National Center for Advancing Translational Sciences, has said that gene editing promises a new era of precision medicine. KJ's story is a powerful early chapter in this new era. While the scientific and medical achievements are astounding, significant questions remain. One of the most pressing is the affordability of such hyper-personalized treatments. Conventional gene therapies already cost millions of dollars. Creating a unique therapy for each individual patient could be even more expensive. Ensuring equitable access to these life-saving innovations will be a major challenge. Society must grapple with how to fund and deliver these treatments. We need to balance the incredible promise of precision medicine with the need for fairness. The journey ahead involves not only scientific discovery but also careful ethical and economic considerations for the future of healthcare.